So most of you will have recognized by now this gentleman here, I suppose. It's Leonhard Stock, of course. And he is the 1982 world champion in downhill skiing, and a fellow Austrian and a big role model and legend. But what you may not know, at least some of you, is that he was never supposed to compete in this race. In fact, he was just a last-minute substitute for Franz Klammer, who was a much more accomplished champion at the time, and he had to cancel last minute due to a minor left knee injury, I believe. And so Leonhard stepped in, and the rest is history. He won the championship. He became a big star and filthy rich. And with this in mind, I was very happy to accept Alain's invitation to join the symposium today. And so I will talk about signal and noise in the olfactory system. And so in my lab, we're interested in how olfactory signals are detected, how they're being processed, and how the brain generates a perception of smell that can drive and control olfactory-driven behaviors. And I will try to convince you today that the olfactory system has evolved exceptionally powerful mechanisms to detect and to amplify very weak signals, and to do that in the presence of a lot of noise in the environment. However, that such mechanisms of amplification and suppression of filtering come at the cost, which may be an increase in the variability or the stability of an olfactory sensory representation in the brain, with potentially important consequences for odor coding and behavior. And so these are the kinds of behaviors. So here is Noam Sobel's student from the Weizmann Institute in Israel, and he is sniffing his way along a path of chocolate trail here, indicated in white towards a source of chocolate. And so as you look at this picture, clearly his student is capable of identifying the smell of chocolate. So he knows, he can tell and discriminate the smell of chocolate from, let's say, the smell of grass. You also appreciate that he's going zigzag, and that's because he can detect and discriminate different concentrations in the smell of chocolate, which eventually allows him to find the source. And most likely is also doing a lot of filtering such that he doesn't lose his track when he comes across an obstacle, let's say dog poop along the way, for example. And so in other words, the olfactory system has to be capable of encoding the identity of an odor, its concentration and concentrations and changes in concentrations, as well as, and do that in a highly unstable, noisy environment where background odors can fluctuate extensively. And so in the lab, we're using genetics, optogenetics, in vivo imaging, and behavioral approaches to try and understand how that works. And we're not studying students, but we're studying mice. And so this is a very simple, brief outline of the organization of the mouse olfactory system. So odors enter here through the nasal cavity. They're detected by sensory neurons in the main olfactory epithelium. These neurons project axons to the olfactory bulb, the first processing station of olfactory information in the brain. And from here, information is transmitted to several higher olfactory centers in the cortex, including piriform, which is thought to underlie learned or associative olfactory behaviors. Now, odor-evoked activity is initiated in the olfactory epithelium by the binding of odorant molecules to receptors, here expressed on the dendrites of these olfactory sensory neurons. The mouse genome contains over 1,000 of these receptors, yet a given neuron expresses only a single receptor from this large gene repertoire. And neurons expressing a given receptor, so here genetically marked in green, these are the P2 expressing neurons, they're distributed all over the place here in the olfactory epithelium, yet they project axons to a single synaptic structure, glomerulus, here in the olfactory bulb. And these projections to the bulb, they're highly stereotyped, they're conserved between individuals, and they thus elicit a topographic map of receptor activation in the brain. So an odor will interact with a subset of receptors, which then will activate subsets of neurons, which in turn then elicit these dispersed and stereotyped patterns of glomerular activity in the bulb, and these are in vivo two-photon imaging experiments. These 3 glomeruli, for example, light up in response to acetophenone, the smell of almond. So in the lab, we're very interested in how the brain actually reads this map. And so when you look at the projection neurons, the mitral and tufted cells that transmit this information to cortical areas, then they will extend a single dendrite into such a glomerulus, but then extend axons to very large regions of the brain, and including the piriform cortex here. And in striking contrast to this tight spatial organization in the olfactory bulb, these projections to the cortex, they're highly dispersed and lack any apparent topographic organization. And consistent with this anatomy, imaging experiments have shown that orders activate these sparse and dispersed intermingled neurons in piriform without topographic organization. Again, very different to this organization of glomerular activity in the olfactory bulb. So the representation of orders undergoes a quite dramatic transformation from the olfactory bulb to the cortex. And to begin and assess how the neural circuit mechanisms that underlie these transformations and the behavioral significance and constraints of such transformations, we decided a few years ago to manipulate or to perturb the glomerular map. And we did that by generating mice with a so-called monoclonal nose, which instead of expressing a repertoire of 1,000 different receptors, largely express a single odorant receptor, the M71 receptor. And this genetic strategy, the expression of the M71 receptor, due to complex feedback mechanisms of gene regulation, results in the suppression of the endogenous odorant receptor gene repertoire, such that now 96% of all sensor neurons express a single receptor. Now, in the olfactory bulb, these M71 projecting fibers, the expressing fibers, they project to the entire olfactory bulb. Somewhat surprisingly, the general organization is maintained compared to controls. And not only that, the very few neurons that express endogenous receptors, so those 4% that still express the endogenous repertoire, appear to form normal projection patterns as shown here. The P2 neurons, again, marked genetically in green. In the wild-type scenario, they project to a single glomerulus. In the transgenic background, there's very few of these green fibers, yet they do project to a single glomerulus at a position that's comparable to control. So as a consequence, a glomerular map is formed that receives, in these transgenic neurons, that receives severely diminished sensory input from the endogenous odorant receptor repertoire, and then superimposed upon this map are these pervasive projections from M71 expressing fibers. And so these changes in the expression of the odorant receptor gene repertoire have obvious and quite dramatic consequences for these patterns of glomerular activity in the olfactory bulb. And so these are two-photon in vivo imaging experiments on top controls. For individual monomolecular odorants, activate these sparse and dispersed stereotype patterns of glomerular activity. Most odorants in these mice do not activate any or don't elicit any detectable activity, and that's consistent with this dramatic downregulation of the odorant receptor gene repertoire. And then in contrast, and somewhat expectedly, acetophenone, which smells of almonds and activates the M71 receptor, now activates every glomerulus. And so what are the consequences now of such a massive perturbation in the olfactory map for olfactory-driven behaviors? So we tested a set of different behaviors. These are innate olfactory-driven behaviors, so instinctive behaviors that mice don't need to learn, mating, for example, and aggression. And in both of these assays, M71 transgenic mice are severely deficient. So, for example, these M71 transgenic males, they do not mate, so the frequency of mounts is severely reduced, and they don't fight to defend their territory. And both of these behaviors strictly rely on olfactory cues. So innate behaviors are severely perturbed. However, we also found that associative olfactory learning is reasonably well intact. And so this is a appetitive go-no-go associative olfactory learning assay where an odor one of two odors is paired with a water reward. And so throughout a given trial, so 20-minute trial, mice initially do not know which odor is paired with a reward, which one isn't, but throughout the trial, then the frequency, they will only lick when they encounter the rewarded odor, and so the frequency of correct licks goes, to above 75–80%. So using this assay, we found that these mice with the monoclonal nose did in fact reasonably well, or surprisingly well. They are perfectly fine to detect ethyl acetate, so discriminate ethyl acetate from air or nothing. They can also discriminate ethyl acetate from citronellol. They can even discriminate very similar odors that to my nose both smell of lemon, so these are enantiomers of plus and minus citronellol. And then they only begin to show deficits when they are asked to discriminate mixtures of such highly similar odors of 1% minus versus a mixture of 0.5% plus, 0.5% minus citronellol. So despite this massive reduction in sensory input, they're capable of detecting and discriminating pretty much any odors. Perhaps most surprisingly, we also found that they do not detect in this assay the M71 receptor ligand acetophenone. So despite this massive activity that acetophenone evokes in the olfactory bulb, these mice fail to discriminate acetophenone from air in these assays. And so to summarize, or at that point, we came up with basically two predictions. One is, well, these mice failed to detect acetophenone, and we postulated that this pervasive activation of the olfactory, of the glomeruli may sort of reflect an extreme genetic scenario of olfactory noise, and this noise may simply be entirely suppressed, okay? And second, these mice can detect and discriminate most odors, and this clearly suggests that there must be mechanisms in place which allow for the amplification of very weak sensory stimuli. So if one looks at the organization of the olfactory bulb, then in fact it's been suggested for a long time that its organization may be well suited to mediate such amplification and suppression or normalization of sensory signals. And so due to, first of all, the convergence of inputting sensory neurons into glomeruli, and then specifically through three types of interneurons, periglomerular cells at the peripheral level of the glomerulus, PV cells recently been identified in the external plexiform layer, and granule cells in the granule cell layer. So in order to now test our questions and directly visualize, so to test whether this pervasive activity can indeed elicit massive inhibition in the olfactory bulb, we thought to directly visualize inhibitory activity, and we did that by using a conditional GChem3 expressing virus, which we injected in the olfactory bulbs of GAT2 Cre transgenic lines, which express Cre specifically inhibitory neurons. So this is shown here in a histological section through the bulb. These periglomerular at the level of the glomerulus inhibitory neurons are here. They sit around the glomerulus. PV cells in the external plexiform layer, granule cells in the granule cell layer. And this is now a top-down view under the two-photon microscope where we specifically focus on the periglomerular cells as they receive direct input from the sensory neurons and can mediate or thought to mediate both pre- and postsynaptic inhibition. And so we can ask, does pervasive glomerular activity inhibit massive inhibition? And the quick answer is yes. So compared to controls, the activity of periglomerular interneurons is massively increased in response to acetophenone. The fraction of responsive neurons, even at very low odor concentration, is very high, above 50%. It further increases as you increase acetophenone concentration, but it very quickly reaches a plateau of about 90% of cells responding to such a stimulus. And again, this is massively increased compared to controls. Not only is the fraction up, also individual responses are highly increased. So these are example traces of such individual periglomerular interneurons in response to, again, 3 different concentrations of acetophenone. And you see that in these transgenic animals, these responses are massively increased. In fact, the scale is different here on the y-axis across these 3 different concentrations of acetophenone. So indeed, pervasive glomerular activity does elicit massive periglomerular inhibition. Now, how does this balance of excitation inhibition result in a mitral cell odor representation, which is sort of the output of the olfactory bulb? In order to address this question, now we had to image mitral cell activity, and we did that by using a modified rabies virus, again expressing GCAMP calcium indicators. We injected rabies into the cortex now, and because rabies specifically enters neurons via nerve termini, we very specifically, if we then look in the bulb, we very specifically label the mitral cells in the olfactory bulb, and this is shown here in a section for the bulb where the mitral cell layer is very specifically labeled. Here at high magnification, you see dendrites innervating a single glomerulus, and then axons projecting to the bulb. And again, a top-down view under the 2-photon microscope where we can expose mice to stimuli and record odor-evoked activity. And so we characterized odor-evoked activity. Such mitral cell responses are very robust and reliable. So again, this is an example of an individual cell responding to different concentrations of acetophenone. They're, again, very robust. They also exhibit a sort of dynamic range with increasing concentration. If we use now a panel of different odorants, we see that mitral cell odor representations are sparse and distributed and relatively specific. So here are 3 different odorants: amyl acetate, acetophenone, farnesene, and a pseudo-colored response map, which is overlaid here in this 4th panel. And so you see that at this low odor concentration, about 10 to 20% of neurons respond to a given stimulus. And we can also look at how these cells are tuned, how specific are these responses. And you see that some cells, for example, are very specific. So this red one will only respond to farnesene, or the green one only to acetophenone, while the blue ones here are cells that respond to multiple odorants. So there's a distribution of tuning, which is again quantified here in this graph. And so the majority of mitral cells, in fact, respond to 1, 2, 3, or 4 odors, and very few are more broadly tuned. And we also characterize response magnitudes, etc., duration. And I should mention that these data are very consistent with recent experiments from the Isaacson and Masrani labs using very similar experimental approaches. So with this in hand, we can now ask, how does this pervasive input and this massive periglomerular inhibition translate into mitral cell odor representation? And we find that indeed at low odor concentrations, mitral cell responses are quite sparse. In fact, they're very similar to what we observe in controls. However, in transgenic mice, when we then increase the concentrations, the fraction of responsive neurons goes up significantly, and at 1% odor concentration, you have massive, widespread mitral cell activity reaching about 80%. So this suggests that indeed, at low odor concentration, inhibition can balance this massive excitation, but that this balance is fragile, and very quickly, as inhibition reaches a plateau, then mitral cell activity is widespread and sort of out of control. However, so inhibition suppresses pervasive olfactory noise, at least at low concentration, but also this naive prediction of ours that simply all activity is suppressed is not true, and we're likely missing additional parameters of mitral cell response properties, which will explain the behavioral deficits we observe in these M71 transgenic mice in response to acetophenone. So now we move to the second question, or the second prediction, that there's a massive amplification of these very weak signals that go into the olfactory bulb of these mice. And for that, we use this 12-odor panel. Most of these odorants, in fact, have been shown to not activate the M71 receptor and consistently do not elicit any detectable glomerular activity in these animals. And we observed that the fraction of responsive neurons in these transgenic animals in response to this large odor panel is very, very similar to what we observe in controls. So about 10 to 20% of neurons respond, despite the fact that we cannot detect any glomerular activity. Consistently, tuning is similar. So again, most odorants respond to only, most cells respond to only a few odorants. In fact, the neurons responsive to a single stimulus is in fact a little bit increased. And so these data clearly show that signals, weak signals, can massively be amplified in the olfactory bulb of these transgenic mice, yielding very robust and reliable mitral cell odor representations at frequencies, tuning, and magnitudes, which I don't show, that are indistinguishable from control. Now, this amplification, we believe, has a cost, and the cost may be the variability of the odor representation. And so while in wild-type animals, mitral cells respond very reliably to an odor stimulus, in transgenic animals, this variability is significantly decreased. And that's shown here in this graph. We see that in control animals across a large set of stimuli, most mitral cells respond 3 or 4 times in repeat stimulations to the same stimulus. And in transgenic animals, the frequency of, or the fraction of cells responding 4 out of 4 times is now significantly reduced, while most mitral cells only respond 2 out of 4 times. And so, to summarize, I've shown you that, so we've characterized glomerular activity, paraglomerular inhibition, and mitral cell output in olfactory bulbs of M71 transgenic mice with a monoclonal nose, in which this map of glomerular activity is massively perturbed. I've shown you that pervasive glomerular activity results in strong paraglomerular inhibition, which, at least at low input strength, can result in sparse mitral cell activity. However, this balance breaks down, is very fragile, breaks down at higher order concentrations and resulting in massive mitral cell activity in these transgenic animals. At the same time, weak olfactory stimuli can robustly be amplified to generate mitral cell responses that in frequency, magnitude, and tuning are indistinguishable from controls. However, this amplification comes with an increase in trial-to-trial variability. And we believe that this variability may be particularly important to discriminate similar odorants or odorant mixtures which have highly overlapping mitral cell odor representations. And if that's the case, then this variability may, in fact, provide a simple explanation for the behavioral deficits we see in these animals with difficult to discriminate odor pairs. Now, more generally, and sort of looking into the future, this variability raises very interesting questions, I believe, with respect to how mitral cell representations are integrated at the next level of odor processing in the piriform cortex. And so very recently we have in the lab generated new genetic imaging and mathematical tools, in fact, to characterize odor representations in the olfactory cortex. I'm not going to talk about them, but I'll show you just one slide to illustrate the massive amount of diversity and variability we see. So these are 15 odorants and 20 odors. Blue are individual responsive trials, and red are average trials, responsive trials, and gray are non-responsive trials. And so as I mentioned, we see a very large degree of variability, heterogeneity, which likely reflects 3 main variables. One is that we have a large variety of different cell types in the cortex with different response properties, etc. Second, these cells are connected densely, both locally and across large distances within the piriform cortex. And third, these responses likely reflect both sensory-driven inputs as well as top-down modulation of neural activity. And so this is what I hope to show you at the next neural circuits meeting. And at this point, I would like to acknowledge the people who have performed the work. So in my lab here, mostly Benjamin Roland, a PhD student, together with Dara Susolski now at UC London. We've been helped by Andreas Schiffen and Sumi Fukunaka, the NMR in London, and Kevin Franks at Duke University. what do you say? Thank you for your attention. I'll be happy to answer questions.